Hello, and welcome to the webinar, Advances in Tissue Engineering, Bioprinting, and Body-on-a-Chip Technologies, an update for regenerative medicine workforce development. I'm Gary Green, and I'm the Chief Workforce Development Officer for the Wake Forest Institute for Regenerative Medicine and Chief Operating Officer for the Regen Med Development Organization. I want to thank our partners in the development of the webinar and our presentation today. First, the Wake Forest Institute for Regenerative Medicine, the Regen Med Development Organization. We will have the AlphaMed Press, which is providing technical support for the development of our webinar and the presentation of our webinar today. And then sponsorship through a grant by the National Science Foundation Advanced Technology Education ATE program and additional financial support from the Bank of America. We appreciate the support of all of these partners. Today's webinar is an intent to bring three of the fast-moving and most exciting components of regenerative medicine to you in order to be able to share this information across the educational spectrum, from community colleges to baccalaureate institutions to graduate programs, but with a particular focus on developing the skilled technical workforce. I want to introduce our speakers for today. First, we have Dr. Tracy Criswell. Dr. Criswell is the Associate Professor at the Wake Forest Institute for Regenerative Medicine, or WFIRM. We also have Dr. Sean Murphy, also Associate Professor at WFIRM. And we'll have Dr. Thomas Shupe, Associate Professor at WFIRM as well. All of these presenters are global leaders in the fields of regenerative medicine, and in particular the technologies that we're addressing today. So with no further ado, I would like to begin our webinar, and we will return after the presentations with live discussion with our presenters. Enjoy the webinar. Thank you.
Hi, my name is Tracy Criswell, and I'm an associate professor at the Wake Forest Institute for Regenerative Medicine. I'm going to give you a short introduction to the field of regenerative medicine and an update on the status of this field today. First, we need to start by defining what we mean by tissue engineering in regenerative medicine. Basically, regenerative medicine is a field of science that combines engineering principles with biology and medicine to enhance, repair, or replace damaged tissues or organs. Regenerative medicine can be thought of as a large umbrella that encompasses tissue engineering and biomaterials, cell therapies, and medical devices and artificial organs, which we refer to enabling technologies. So tissue engineering, the idea of tissue engineering is really building an organ or tissue outside the body to replace a damaged or diseased organ inside the body. So it's an inside or it's an outside in type of approach, where cell therapies really are addressing the ability of a tissue to heal itself. So more of an inside approach, where we use cells to either aid in repairing a damaged tissue, or we use cells to provide factors that a damaged tissue needs to heal itself. So the need for this field. Well, regenerative medicine has the potential to affect many different areas of human health and disease. And if you just look at this image, you can see there's the runner with an amputation. There's, victims of car accidents. There's the baby with the cleft palate. Up here, you can see there's scars going to form at the site of an injury, right? And so regenerative medicine can be used to address all of these different human health issues. In addition, if you look at this graph, all you need to look at is the gray and the green lines, right? So the gray line indicates the number of patients waiting for an organ transplant. whereas the green line indicates the number of donors available, of organ donors. And this is an older graph, only goes up to 2009. This discrepancy between these two, the need and the supply, has grown greater over the past decade. And so tissue engineering would allow us to engineer new tissues for people that need an organ transplant, and we could then, close this discrepancy. So this, most of you may have seen this before, but this is a newt or salamander, and they have a really remarkable ability in that if you cut off their leg, it will grow back and it will be identical in form and function to the one that you removed. So the question is, why can't we do that, right? Now, with that said, most tissues in our body do have a regenerative capacity. They are able to deal with minor injuries. And so this is a paper cut. We've all had paper cuts, they really hurt, but within a couple of days, it's healed and gone. Now, on the other hand, our bodies cannot handle large injuries. This is an ulcer that you see below. We can't heal those types of tissue damage. And what happens is we end up with loss of function. If we're able to retain the limb, if we don't lose the limb, we end up with loss of function and scar formation. And so tissue engineering provides an opportunity to heal these types of larger injuries. So that leads us to, what do we need to engineer tissues and organs? Well, there's three basic components that we use when we talk about tissue engineering and regenerative medicine. This would be cells, scaffolds, and signals. And I'm going to talk about each of these briefly. We're going to start with scaffolds. So this is a house. We all know that this is going to be a house. It looks like a house. Parts of it might function as a house. It's got kind of a roof and some windows, but it doesn't function properly as a house. You could live there probably with a sleeping bag, but if it rains, you're going to get wet. If it's cold, you're going to be cold. If a raccoon comes by, it's going to walk right into your house, right? So this is the form of the house, but it doesn't provide the function of the house. And when we talk about scaffolds, that's what we mean. A scaffold provides the form and structure of the tissue or organ that we want to engineer. And so scaffolds can be synthetic sources, so these are man-made sources, or they can be naturally available sources. And the type of scaffold that you use is going to depend on the tissue that you want to engineer. So if you look at the top, this is a hydrogel scaffold. So it's a soft scaffold. It's soft. It's aqueous. It's got water in it. It's porous. And so this type of scaffold we would not use, obviously, to make a bone. Bones need to be strong, supporting, weight supporting in some instances. This type of scaffold might be used, say, for cornea. Now, if we want to make skin, we're going to use a flat scaffold, right? And then if we want to make a blood vessels are essentially tubes, right? So we can take that flat scaffold and we can wrap it up into a tube and use that for engineering blood vessels. The other technique that we use to develop scaffolds are decellularized organs. And you can see that at the bottom here. This is a rat liver and a rat muscle that have been decellularized. Now, what do we mean by that? We mean that the rat liver has been removed from the animal. We have used different gradients of detergents or soap, essentially, to remove all of the biological material. So all of the cells come out, hence decellularized. And once we remove all of those tissues, what we're left with is the scaffold of the liver or the tissue that we've decellularized. And so you can imagine that we could then take that scaffold and put healthy cells back into it, right? That would provide the exact form that's needed for function of a healthy tissue that could go into a patient. So we're going to move to cells. We use stem cells, and we're going to define what we mean by stem cell first. So a stem cell has three characteristics. One, it can repopulate the stem cell pool. Now, what do we mean by that? Well, the stem cell pool is the number of stem cells that exist in a tissue. And so I'm going to use skeletal muscle as an example because of the tissue that I work with primarily. Skeletal muscle has a pool of stem cells. We call them satellite cells. When you injure your muscle, those stem cells start to divide, and then they move to that area of injury, and then they differentiate and become new muscle tissue. Now, if the stem cell did not repopulate itself, the next time I injured my muscle, it wouldn't be able to heal. And so that's what we mean by repopulating the stem cell pool. To do that, we have the second characteristic of stem cells. They undergo asymmetric division, meaning that one cell can become either another stem cell, and it can also become, it can become a precursor cell to the specific tissue it's going to differentiate into. And the third characteristic is that stem cells are potent. And what do we mean by potent? Potency refers to the ability of a stem cell to become other tissues. And so there's different types of potency. A totipotent stem cell, as you can see at the top, a totipotent stem cell is essentially derived from when an oocyte or an egg is fertilized by the sperm. It's a very early zygote or embryo. This type of cell can become all the tissues in the embryo, as well as all the extra embryonic tissues, such as the placenta. The next step is the pluripotent stem cell. So the pluripotent stem cell is what, when we talk about embryonic stem cells, these are pluripotent stem cells. These are also induced pluripotent stem cells, referred to this stage of stem cells, this potency. These cells can become all the tissues of the body. So these are derived from the blastocyst, which is still an early zygote during development prior to implantation into the uterus. So these cells can become all the tissues of the body, but they don't form the extra embryonic tissues. The third step down would be the multipotent stem cells. These cells are restricted in differentiating into a specific tissue lineage, which you can see over here, either ectoderm, mesoderm, or endoderm. And you may have heard of mesenchymal stem cells. They're the most commonly used stem cell in tissue engineering and regenerative medicine therapies. And then the next level down is the unipotent stem cell. These are tissue-specific adult stem cells. These are the stem cells that become activated when you injure your tissue, like the satellite cells for skeletal muscle that I described in the previous slide. So where do the cells come from? And this is actually one of a major issue that we face in tissue engineering and regenerative medicine today, is trying to find a good source of cells, right? So when we talk about transplantations or cell therapies, we usually talk about autologous or allogeneic. An autologous source means cells from the patient would go back to the patient. An allogeneic source means cells from a donor would go into a patient. And so autologous is ideal. Allogeneic is what we're usually left with. And patients with an allogeneic transplant or stem cell injection have to go on immune suppressive drugs so they don't reject the graft. And most of our tissues have, as I said before, have a source of stem cells, the unipotent or adult stem cells. But when we talk about cell therapy, in general, the allogeneic stem cells come from donor discarded operating room tissue or from a cadaver. So neither one are really an ideal source of stem cells. So this is something that we're working on. How can we isolate and then expand enough stem cells to use in tissue engineering therapies? The last thing we're going to talk about is cell signaling. And so I really like this cartoon up here. I thought about putting in an image where, in class, we're passing notes to one another. And then I realized, well, I bet people don't really do that anymore because we all have phones. And so we talk to each other, right, through our wireless devices. And so we're talking to each other, we're signaling to each other. Well, the same thing happens with cells. Cells talk to each other, and they can talk to themselves, which I do quite a bit, unfortunately. And we would call that autocrine signaling. Cells can talk to their neighbors, which would be paracrine signaling. And then cells can talk to cells that are in another tissue by signaling through the bloodstream. And that we call endocrine signaling. And so cell signaling is very important. Cells do not exist in a vacuum, they exist in a tissue. And so once we take those cells out of the tissue, we need to come up with ways of providing the right signals to keep them alive, to keep them happy, to keep them growing, and to help them differentiate into what they're supposed to be. And so where are we today? Well, we're working on the whole spectrum of organs. In the clinic, we're still pretty early on. Flat structures, which is cornea, skin. Tube, remember the blood vessels are really a flat structure that you can roll up into a tube. And then you've got your hollow structures, which are also basically a flat structure that are, balled up into like a balloon-like structure. The reason why this is where we are is because these are very thin. And so we started by saying that cells require oxygen and nutrients to stay alive. Oxygen can only diffuse about 3 millimeters. So all of the structures right now that we can engineer up to a size that can be used in a patient have to be very thin. So the problem we're facing here is how do we integrate a functional vasculature that would allow us to feed the cells in these solid organs, such as kidney, liver, pancreas, skeletal muscle. How do we grow an organ or engineer an organ while maintaining the viability of the cells on the inside of that organ as we grow it large enough to function? So I'm going to end there. Thank you very much for your attention.
Hi, everybody. My name is Sean Murphy. I'm an associate professor at the Wake Forest Institute for Regenerative Medicine. Today, I'm going to be presenting on 3D bioprinting and its applications for airway tissue transplantation. So I'd like to start off with a definition. 3D bioprinting is the combination of living cells, biomaterials, and 3D printing technology to develop a complex living tissues with physiological structure and function. Many of us are already aware of 3D printing technology as it's been around for many years, primarily in the rapid prototyping or additive manufacturing stage, where non-biological materials have been used to develop very complex three-dimensional structures with a range of really important properties. And 3D bioprinting follows the same principles, and it's basically the same approach of depositing materials in this layer-by-layer fashion to build up a complex three-dimensional structure. So today I'm going to provide a brief overview of the 3D bioprinting process using some examples from my own lab. In a 15-minute webinar, I don't really have the time to go into the detail of every type of material or every bioprinting modality that exists. So instead, I'm going to just focus on some of the core principles and explain how we've used them in our lab for one application. So overall, the general process of 3D bioprinting usually begins with some sort of imaging modality, whether it's CT or MRI or X-ray. This information is fed into a computer-assisted design or manufacturing type software to aid in the design and the development of your construct. The biomaterial and cell selection is probably the most important component of 3D bioprinting. There's a whole range of different types of biomaterials or bioinks and different types of cells that you can use to match your application. Probably the most fun part of this process is using the bioprinter itself. There's a range of different types of bioprinters, and they're constantly developing, ranging from extrusion bioprinting, coaxial, digital light processing, and many more. This is a really fast-moving area. There's a couple of optional steps after you actually bioprint your functional tissue. You can place them into a bioreactor to exert physiological forces on your construct and get them mature and ready for transplantation. And an alternative to implantation, many groups are using 3D bioprinting to build functional tissues for in vitro testing. So there's a whole range of different applications here. It's not just for transplantation. So today I'm going to try and talk about all of these different components using an example from my own lab where we've been attempting to develop a 3D bioprinted trachea. So as a summary of what I'm going to talk about, we used a CT imaging modality with computer-assisted design to develop our trachea. We actually used autologous mesenchymal stem cells as our cell source, and two different types of materials, which I'm going to break down into generally a functional and a structural type of material. We used an extrusion bioprinter, which has been developed at Wake Forest. And our final goal application for our 3D bioprinted trachea was for transplantation. So why do we need to develop new methods for tracheal reconstruction? Well, I was quite shocked when I started this work to find that the current method of tracheal reconstruction was a formalin fixed allograft. So these are donor tracheal tissues which are actually formalin fixed before they're transplanted. So these tissues are no longer living. They have the right structure and the right size and shape as our tracheas, but as they're formalin fixed, they're not going to grow with the tissue with the patient. They're not going to remodel. And so there's some serious limitations there implanting a fixed non-living tissue into a patient. For this reason, there's been a range of experimental methods developed, including xenotransplantation, decellularization, different types of tracheal stents, or 3D printed splints or molds. But these have all got their own limitations. They have some strengths, but most of the time, these limitations mean that they've not been successful for long-term transplantation. So our overall goal was to develop a patient-matched 3D bioprinted tracheal tissue, which has been suitable for transplantation. There are two main functionalities or two main sub-goals here that we focused on. The first was that I wanted to have autologous tissue. This means it's the patient's own tissue to prevent rejection and not require the need for immunosuppression. Another advantage is that this would facilitate remodeling over the long term. So after transplanting the patient's own tissue, this will grow and be with the patient for the rest of their lives. However, this does require an easily accessible patient cell source. The next component, and probably the most challenging, was what I call a day zero functionality. So many of the patients that require emergency trachea reconstruction are in the hospital for emergency trauma. So these could be soldiers which have been exposed to an IED or gunshot wounds or other similar types of emergency trauma where the patient simply can't wait for many weeks or months for us to develop these tissues. So this means that we couldn't use a bioreactor to try and mature these tissues before we implant. Most of the tissue development was required to occur in vivo. So to take a step back, looking a little bit of what the actual structure of the trachea is and what we're trying to reproduce, we first of all took some of this formalin fixed allograft tissue, and shown on the left is a human tracheal tissue. And you can see from this cross-section that the tissue is really formed up by a C-shaped cartilage ring, which is held together by a series of smooth muscle tissue. This holds this cartilage rings together as well as sits against your esophagus to provide flexibility to move your head and your neck, as well as to swallow food without blocking your airway. Of course, this is all lined by the tracheal and airway epithelium. So we took this tissue and performed a CT scan. And from that scan, we performed a series of measurements looking at the inner diameter, the cartilage shape, structure, width, and association to each other. And we took all of these measurements and fed this into our computer-assisted design software. And our overall goal was to replicate a subsection of the tissue representing 3 cartilage rings and the associated smooth muscle tissue. This brief video just shows how we built this tissue in this layer-by-layer fashion using our software, with the green lines representing the toolpath that will eventually build the cartilage rings, and the red representing the toolpath that's going to build the smooth muscle tissue. You can see this is a relatively simple design representing these 3 cartilage rings and the associated smooth muscle tissue. Next, I'm going to give an overview of our 3D bioprinter. This is developed at the Institute for Regenerative Medicine, and we have several versions of this, but this is the one that we used. First of all, we have a series of print heads. This specific printer uses 6 print heads. In our experiments, we just used 3 of them, one each to print cartilage tissue and smooth muscle tissue, and a third one to print a PCL, which is our structural material, which I'll get into in the next few slides. We have an XYZ controllable stage, and this is able to move in 3 dimensions to allow the tissue to be printed, but also to grow in 3 dimensions. We have pneumatic solenoids, which control the deposition of each of the bioinks, as well as temperature and pressure control modules, which control the environment during printing. And finally, we have an air control system, which is used to keep this entire area sterile during printing. So moving on to the biomaterials that we chose. To start with, I would like to talk a little bit about the scaffold material, or polycaprolactone. This is a semi-crystalline polymer with a 60-degree melting point. So this is ideal for the melt extrusion print head that we have in our bioprinter. It's biodegradable and biocompatible, so we can implant this into the patients, and over a series of months to years, this will eventually break down and allow the formation of the patient's own tissue. For many reasons, this polycaprolactone has material properties which is ideal as a scaffolding type material. Primarily, we can build relatively complex 3-dimensional shapes. It has the strength and stiffness to be able to resist pressures, and replicates very similarly the mechanical properties of the airway tissue. And this is just an example of some of the different types of mechanical testing we performed on structures built using polycaprolactone. So we tried to apply the strength of this scaffold biomaterial to try and develop a tracheal structure which very closely matched that of the donor trachea. So as I mentioned before, there are 2 main regions that we're trying to replicate. First, the cartilage region, and second, the membrane or the smooth muscle region. So we developed a few different designs. We had one design for the cartilage region, as shown on the top there. We had 4 different designs for this smooth muscle region, which sits against the esophagus and provides the flexibility. And our goal was to print these designs, perform mechanical testing, and test them against the donor trachea to see which ones matched best. So we performed tensile, yield stress, and elastic modulus testing of our 3D printed PCL cartilage region compared to the donor trachea. The donor trachea is shown in red, and our printed cartilage region is shown in black. And you can see that this first attempt at printing the cartilage demonstrated that our print was successful, and that our yield stress and elastic modulus was very closely matching that of the human trachea. So we're very happy with this first initial design, and no further iterations were performed. When it came to the membrane region, we weren't so lucky. It just took a little bit more work. We originally ruled out design 1 initially, just because we simply were not able to seed cells in any of the pores with this design. So we didn't even perform the testing there. So we limited our testing to designs 2, 3, and 4 shown on this slide. And as you can see from the yield stress and elastic modulus testing, we found that design 2 was not very good. It had a much higher tensile yield stress and elastic modulus compared to the donor tracheal region. But designs 3 and 4 were really close. While design 4 was probably the closest, we actually favored design 3. And the reason is that this had a lot more space and a lot more porous areas to allow us to incorporate some of the functional biomaterials, which are going to actually encapsulate our cells and eventually differentiate into functional tissue. So we ended up selecting this cartilage region and design 3 of our smooth muscle region. So that's the structural material and how we decided to design and select our materials for the structural components of our tissue. However, the structural component by itself is too porous. Cells do not like to grow on it, and it has some other limitations which require us to also utilize a functional biomaterial. And in this case, we used a hyaluronic acid-based biomaterial. So this bioink consists of dilated hyaluronic acid, dilated gelatin, and a crosslinker, which is called PEG-D malleamide. The advantage of this bioink is that these materials are naturally occurring and found in quite high concentrations in cartilage tissue. It's been previously used extensively to support 3D cell culture, and our studies have shown that it doesn't induce any immune response. It doesn't have any toxic degradation byproducts, and it's actually really useful to have shear thinning properties, which allow us to use our extrusion bioprinter to deposit this material. And as I'm going to talk about in the next few slides, it has tunable properties which enable us to drive cell differentiation. So what do I mean by tunable hydrogel properties? Well, by taking that PEG-D malleamide crosslinker and adjusting simply how many arms it has, whether it's got 2 arms, a linear crosslinker, 4 arms, or 8 arms, we can actually adjust the porosity of this bioink after it's printed. And what does that mean? So that means with a lower pore size, we actually increase the stiffness of this hydrogel. This allows us to have a whole range of different stiffnesses, ranging from very soft to relatively stiff. It also has the advantage is in that it allows us to load growth factors into the hydrogel, which are released over a relatively long period of time. And this is going to be very important for the maturation of these tissues in the long term. So moving on to the cell component, as I mentioned earlier, the cells that we decided to use for this 3D printed trachea were autologous bone marrow-derived MSCs. There are many reasons for choosing the cell type. The primary reason is that this is an easy to access type of cell type, either through bone marrow biopsy or even from adipose-derived tissue. We can isolate this from the patient, isolate the cells, grow them, and implant them in a relatively short period of time. A second advantage of MSCs is that they're known to differentiate into multiple lineages, including chondrocytes and myoblasts. So chondrocytes are the cell types which form cartilage tissue, and myoblasts are the precursors to smooth muscle tissue. Importantly, other groups have previously shown that just simply by adjusting the elasticity or the stiffness of the material that these cells are embedded in can actually drive the differentiation into these tissues. So slightly softer hydrogel will push MSCs into a chondrocyte lineage, a slightly stiffer gel pushes them into a myoblast lineage. So we were able to utilize this property combined with growth factors to take a single cell source, these MSCs, bioprint them in a very similar gel and push them either towards chondrocytes or towards myoblasts. And this slide really just shows a confirmation of that approach. We're able to take our MSCs, embed them in what we call our smooth muscle gel, which is a slightly stiffer version of this gel, loaded with TGF-beta1. And after a short period of time, these cells were able to differentiate into smooth muscle and actually showed much better differentiation within the hydrogels compared to standard plastic differentiation. A similar approach was shown using the same cells with our cartilage gel, slightly softer and different growth factors, but still able to differentiate into cartilage over a period of time. So this really demonstrated that we could take a single cell, embed it in our gel, and differentiate into 2 different types of tissues that we would like to generate. This next slide just shows an example, a video of the 3D printed tissue. What I'm showing you right now is the polycaprolactone scaffolding material being extruded using those designs I showed you earlier. After each layer of PCL being printed, this is followed behind by a hydrogel containing cells using the different material depending on whether we want to print cartilage or whether we want to print smooth muscle. And you can see this is built up in this layer by layer fashion using that same template I showed you in the previous slide. And this is what the final product looks like with these C-shaped cartilage rings and associated smooth muscle tissue. So we still have a long way to go, and this is very preliminary work, but I hope it was useful to give you an idea of how we're combining these different components of 3D printing to develop functional tissue. Some of our ongoing work includes subcutaneous implantation of these constructs to evaluate how well these cells actually form mature cartilage and smooth muscle, making sure that they actually form healthy mature tissue over time and that the degradation occurs at an appropriate rate. Of course, once we can show that, the next step is to move forward into small animal models, large animal models, and then hopefully into human patients. And we've published some of this work previously. There's still many challenges involved in this approach, and we've faced some of these in this exact project. One major challenge is cell sourcing. I've talked a lot about MSCs, but one limitation from what we're doing right now is that we don't have a good autologous epithelial cell source. We're able to take a very small biopsy of these cells and grow them in the lab, but to generate a very large number of cells will take a long time, and we simply can't take a large biopsy from these patients. So we've been working on a technique called conditional reprogramming, which is a way to use small molecules to very rapidly expand a small number of cells that would be suitable for bioprinting. The next challenge is in our bioink properties. As you see, we currently use a compromise between multiple materials, some for scaffolding, some for function. So our group is working on what we call a universal bioink that can have the best of both worlds with customizable properties for different types of applications. And finally, as we look to build bigger and larger tissues for transplantation, we're going to come across challenges with hypoxia because these tissues are not yet fully vascularized. So to try and overcome this challenge, we're working on bioinks that can both suppress metabolism to buy us time for vascular development to occur, as well as bioinks that can actually generate oxygen to keep these cells happy during this vascularization process. So with that, I'd like to thank you all for your time and thank many of the group here at WFOAM who've worked on this project to make this possible. So look forward to speaking with you all soon. Thank you.
Hello, everyone, and I'd like to thank the organizers for the opportunity to present today. My name is Tom Shupe. I'm an associate professor at Wake Forest Institute for Regenerative Medicine, and I'm one of the technical leads on our Body on a Chip program. We used to call the program the Ex Vivo Console of Human Organoids, or ECHO. We've sort of gotten away from this. We don't like the word organoid so much anymore and use the term organ tissue equivalent, sort of to better differentiate ourselves from groups that have less sophisticated systems. Anyway, let's get started. So our Body on a Chip program was started really to address some of the deficiencies of both animal models and two-dimensional cell cultures in terms of modeling human physiology and pathophysiology. Animals are notoriously poor at mimicking the response of humans in terms of responses to drug toxicities and certain diseases. Animals are expensive and slow, and we're trying to move away from animals as much as possible and come up with new alternatives. 2D cell cultures, as soon as you take cells outside of the human body and put them in cell culture, they have to adapt to a very foreign environment. They change their phenotype, and the responses you get from those cells are drastically different than what you'd expect. So the overall goals of the program, we knew we wanted to build a three-dimensional construct using a lot of the clinical scale tissue engineering principles that we've developed here at the Institute and use them at the micro scale. We wanted to use human primary cells, so we're not using cell lines here, we're using cells from an actual human body. We wanted the system to be integratable, so we wanted multiple tissue types to be represented on one chip. And the goal, of course, was to accurately model the effects of drugs and other human diseases. So a bit about design strategy. What we knew we wanted to do was take as many of the components of the normal cellular microenvironment and put them in culture in our system to reduce the need of the cells to adapt to a foreign microenvironment, hopefully hold that phenotype closer to a physiologically relevant one, and get better responses from the tests that we do on the OTEs, or the organ tissue equivalents. So the three major driving microenvironmental components that we've engineered into the system are the three-dimensional structure. With 3 dimensions, you get that good paracrine and autocrine signaling that will allow the cells to understand that they're in a normal tissue, understand they're in a normal environment, and express a normal cellular phenotype. We wanted to include all the relevant cell types in each model. So for example, here we have a liver OTE. We've got hepatocytes and biliary epithelial cells. We also have fibroblasts, epithelial cells, and we include macrophages so we can provide an inflammatory response to these tissues. The second major factor that we used, we knew we wanted to have a relevant extracellular matrix. And our institutes pioneered a lot of the steps, or a lot of the methods for decellularizing intact organs. We can decellularize a native tissue type, get that extracellular matrix, solubilize it, incorporate it into a hydrogel stroma of some sort. Here's an example without and with extracellular matrix. These are human primary hepatocytes after 7 days of culture. When you don't include the extracellular matrix, all the cells are dead, whereas they remain alive when you have extracellular matrix. Tunable mechanical stiffness. We knew we wanted to have a normal mechanical microenvironment for these cells, so we've developed a lot of cell substrates that we can embed these OTEs in, and through some chemistries that we've developed, we've been able to impart a desired stiffness on that substrate. So You can see we can range from soft tissues like brain all the way up to more firm tissues like muscle. The OTs sense that mechanical environment, and it helps to support the phenotype that we're looking for in these constructs. So a couple examples of the OTs that we've developed so far. Here's our liver OTs, a spherical structure about 250 microns in diameter. You can see here's a representative serum protein that the liver normally produces, albumin. And you can see after about a week of maturation, they produce a pretty steady state level of albumin. We see the same thing with other serum biomarkers like urea, ceruloplasmin, transferrin, all of the plasma proteins that the liver is known to secrete. We also maintain good xenobiotic or a foreign compound metabolism or detoxification. Here we can see a drug, diazepam or Valium. You can see the gray bar here. We get a little bit of metabolism in the first week, and then we lose it completely in the 2D cultures. Whereas with our liver OTEs, we maintain good metabolism of that drug and get the expected metabolite over the course of a month. Our cardiac OTs live a little bit longer. They'll live actually out to 6 months or so. They beat spontaneously. You can see here a wave of depolarization occurring across the OTE. It always starts at one pole of the OTE. That's because there's a group of undifferentiated cells that stay there and act as a pacemaker. They initiate the polarization, and then the other cells follow suit, and a wave of depolarization occurs across the structure. The beat rate's about 60 beats per minute. We can speed it up with certain drugs like isoproterenol. We can slow it down with other drugs. We're showing a good physiologically relevant response. Blood vessel, we've got several blood vessel constructs. This was one that was developed in my laboratory. You can see here we've got basically a layer of endothelial cells, blood vessel cells that are cultured on top of a semi-porous membrane with a hydrogel stroma below it that we can put different OTEs in. In this case, we use liver OTEs. This is sort of to model the interaction of tissues with the vasculature across the capillary bed. You can see here some confocal imaging showing the proximity of the liver OTE to the blue, which is the vascular monolayer, and we get good functionality out of these. Here's histamine. That's the signaling molecule that opens up the endothelium or the blood vessels, makes you have a runny nose, whatever. We can see here that, and this is electrical resistance across the construct. So when we add histamine, it loosens up the connection between those cells, and you decrease the electrical resistance. If we wash out, we cover that tight electrical resistance across the vascular monolayer. Here's a more complicated planar structure. This was developed by Sean Murphy. He was another one of the presenters in this session. Anyway, we've got pulmonary epithelial cells that sit on top of, again, a hydrogel stroma. We're able to embed inflammatory cells and fibroblasts in that stroma. That sits on top of a blood vessel or endothelial layer. And we've got a fluid circuit below that you can put more cells in, like monocytes or neutrophils. With this system, it's an air-liquid interface. So we have air flowing across the top. It causes a lot of terminal differentiation in the pulmonary epithelial cells, allows them to ciliate, allows them to form a relevant mucus layer, which is important for modeling infectious disease and such. You can see here we've got good histology. Here's our 2D culture, not doing too much. Here's our 3D OTEs. You can see a much more robust and more physiologically relevant histology. Two more types real quick. We've got a blood-brain barrier with a neuronal component, sort of mimic the brain. The neuron component is completely encased in brain microvascular endothelial cells. You can see here the tight junctions holding those cells together tightly on the surface, modeling that exclusion barrier to the neurons on the inside that you see in a normal blood-brain barrier. You can see here we've got a fluorescently labeled protein that does not gain access to the inside of the OTE for that fluorescently labeled protein. Once again, with histamine, if we open up that blood-brain barrier, that fluorescently labeled protein is able to get inside and you can detect it. Another example here, this bar here is our blood-brain barrier OTE, it being exposed to mercury. Mercury is neurotoxic. If we disrupt that blood-brain barrier, the mercury gets inside and kills the neurons inside. We can model that on chip. Final one, testes. We also have ovaries, so we can masculinize or feminize our systems. Our testes OTE produces androgen. Also shows the first signs of spermatogenesis. So there's some hope that someday that someone that's sustained a bad injury and can't produce sperm, we would be able to, maybe from an iPS cell derived from that patient, produce sperm on chip, and they'd be able to have a natural child they wouldn't normally be able to have. So far, we've got a whole host of OTs that we've developed, and we've been able to integrate them together into multi-OT systems, and they're all very well characterized. We've got another group that's fairly well characterized, but we haven't integrated them yet. We're in the process of doing that. So a couple of studies to show their functionality. Here's our liver OTs, and this is showing Tylenol toxicity. You know, a little bit of Tylenol will cure your headache. A lot of it will kill your liver. So here we show that at 0, 1, and 10 millimolar acetaminophen or Tylenol, we show very little toxicity with 1 millimolar, and at 10 millimolar, we lose viability. Here reported by albumin production, we lose viability very quickly. And the 10 millimolar is known to be toxic in humans, so we're able to show toxicity at a relevant concentration of what you'd expect to see toxicity in human serum. Here's, instead of albumin, we're showing the same thing with urea, dose response curve with the 10 millimolar producing the most toxicity. We also looked at clinical countermeasures. So in this case, we're looking at lactate dehydrogenase and alpha glutathione transferase. These are cytoplasmic proteins that are meant to stay inside the cell. When the cells get sick and die, it's released to the outside. You can detect them. That's how we're judging toxicity here. You can see with our 10 millimolar dose of acetaminophen, you get this burst release of lactate dehydrogenase with the acetaminophen. When we add the clinical countermeasure and acetylcysteine, we don't see that release of LDH, and that's indicating that the clinical countermeasure is protecting the OTs, much like it would protect the patient in a clinical study. A couple other real-world examples of the utility of these OTs. Here's 2 FDA withdrawn drugs. These are drugs that went through preclinical trials, clinical trials, were released to market, ended up killing patients, had to be withdrawn from market. We wanted to challenge ourselves, can we detect toxicity in these associated with these compounds? The glitazone was shown to have hepatotoxicity or liver toxicity. We showed dose response here with the red being dead, increase in dead cells. We showed the mechanism by which those cells are dying matched what we saw in the clinical situation. Here's another drug, estemazole. This was an antihistamine that was released and demonstrated cardiac toxicity after being released to market. We showed at very high doses overt toxicity. What was interesting, though, is that we're showing at 10 micromolar, we're showing toxicity. But at 10 nanomolar, we're showing changes in the beat kinetics. What that's telling us is that the functional output, the beating of that cardiac OTE, is foreshadowing problems that might occur at a higher dose or at a longer exposure time. So we really like that cardiac OTE as being able to predict toxicities. Here's a host of environmental toxins, some heavy metals, glyphosate, which is the active ingredient in Roundup that you've seen on TV, has been associated with some tumors. Anyway, we're showing toxicity associated with these environmental compounds that are known to be toxic to humans. We also showed no toxicity associated with compounds that have low or no toxicity in humans. Things like ibuprofen, vitamin C, things that we know aren't toxic, don't give us positive hits, which is important for an in vitro model. Some integrated studies we did. I like this experiment a lot. This one is basically showing baseline cardiac beat rate exposed to epinephrine. As expected, we get an increase in the beat rate of the cardiac OTEs. If we treat that OT with beta blocker, propanolol, we decrease the responsiveness to the epinephrine. So the beta blocker makes it so the epinephrine doesn't increase the heartbeat rate. If we open up a circuit to a liver OT, though, that liver starts to metabolize the propanolol, and lo and behold, we see an increase in the beat rate as that propanolol is metabolized. The OT regains responsiveness, the cardiac OT regains responsiveness to the epinephrine, and we see an increase. This is just a slide showing that we got the expected metabolite from the propanolol, proving that it was the liver OT that was metabolizing the drug. Here's a final integrated system where we've got capecitabine, which is a prodrug for 5-fluorouracil, chemotherapeutic drug, but also shows a lot of off-target toxicity. When we include the liver OT, we get downstream target or off-target toxicity from that activated prodrug. When we remove the liver OT right before adding the capecitabine, we don't see that downstream toxicity because we're not activating the prodrug to the drug that's causing the toxicity. Couple of disease models. Here's a little polysaccharide induced fibrosis in our liver OTs. You can see the green is the collagen. Again, we treat with a clinical countermeasure. This is a small molecule drug that is anti-fibrotic, and we show that with increasing doses of that drug, we're able to hold that fibrosis level down. A couple other infectious disease models. Here's respiratory syncytial virus, our lung OT, add the virus, it gets sick, the histology's ablated, and the inflammatory cells accumulate on the surface of that OT. Here's a bacteria spread. I think this is Pseudomonas, a bacteria spread on the OT over time, showing that infections will propagate and get worse. Here's some work we did with COVID, SARS-CoV-2, showing the spike protein, which is the receptor that the virus uses to gain access to the cells on the surface of the OT. When we ran the experiments, the rate of infection was slowed in our OT compared to 2D. This is because you have a lot of other obstacles to get through. You've got that mucus layer, you've got a 3D structure. It's a more complicated system, and it more accurately models the kinetics of that infection spread. We've got two large programs that we're currently working on, both involving the lung OT. These are multi-million-dollar programs. One is to model chlorine gas toxicity associated with an industrial accident or biowarfare situation, and we're developing countermeasures based on our response of our OTs to these chlorine gas. The other large one we're doing is an infectious disease study using artificial intelligence. In this, we expose the lung OTs to different pulmonary infections, get a lot of data back as to the changes that those infectious agents cause in the OTE, feed that into the AI, and start to teach the AI what the infection looks like, what different infections look like. And the hope is that the AI will eventually be able to predict for an unknown infection what clinical countermeasure might be effective. Pretty much everybody at the Institute's worked on this program. I've got just a few of them here. Thank you very much for your attention.
I'll ask first question. I'll ask this to each of the three of you, and that is, since many of our attendees are teaching faculty, perhaps in addition to research responsibilities and such, but could you share your thoughts on the core or entry-level technical knowledge and skills needed for research and clinical translation in the areas you've been discussing, or in regenerative medicine in general? And Dr. Criswell, I'll let you start with that.
Sure, that's a great question. The field of tissue engineering is vast, and so it actually covers many disciplines. And so my first piece of advice to people that are interested in getting into the field is to figure out what you enjoy doing, and then study that area, because you can integrate that into any aspect of regenerative medicine. More specifically, work on your lab technique skills, pipetting, learn some basic biology and tissue culture. What we're seeing now is that people that have some sort of understanding of computer skills, especially because of the machine learning and the AI that's coming up, you can integrate that into the field, then you'll be highly marketable to many different companies.
Dr. Shupe, your thoughts?
When I think of regenerative medicine and tissue engineering, I think of cells, scaffolds, and materials, and those are sort of the three buckets where I would focus my training. Cells, you need the ability to expand cells out of a human body to a sufficient number to do whatever it is you want to do it with. That's tricky. All our work's been done in cell lines, which grow like weeds, and now you're working with the real thing. Biomaterials, so polymer sciences and chemistries and things like that would allow you to help build scaffolds. And biomaterials, it's kind of interesting because all of the biomaterials we've designed medically were designed not to interact with the human body because we wanted to implant them as braces and rods and screws, and now we want biology to interact with that, so it's a whole different type of chemistry, and materials need to be standardized if you want to bring this thing to prime time in manufacturing. And keep your mind open, whichever of those three buckets you're in, you have to know enough of the other two in order to be able to integrate that all into an idea.
Right. Thank you. And Dr. Murphy, your thoughts
sure.
on those skills?
I think, as you've heard from the other two speakers, our field is highly multidisciplinary. So, you can't be an expert in everything, but I think it's very important to learn enough about all of the different topics, ranging from the actual clinical need in medicine all the way to mathematics, software, and everything in between. So you can at least speak the language and understand how all of these different fields interact with each other.
Great. Thank you. And Dr. Murphy, I'm going to come back to you with this next question too. It's a more of a technical question from one of our attendees, but what are the key parameters for the formulation of a bioink? Do they depend mainly on the type of cells in the ink or the tissue we intend to regenerate, or on other factors?
Sure. Yeah, a bit of both, of course. They're both essential. I like to think of it in three different ways. And of course, there's more than three. But these are the three main things I think of. First of all, for it to be a successful bioink, it needs to be printable. So you need to think about what type of printer that you have, whether extrusion or laser transfer or digital light printer, and make sure it's compatible with that. The second, it has to be compatible with the cell type, both being able to print your cells and keep them alive, but also drive their function after printing. And the final part is the time component. What is it going to be over time? These bioinks have to start off generally as a liquid and be printable, but over long term, after you transplant them, you actually want them to function as the tissue. So these bioinks actually have to become part of the tissue eventually and function as part of the tissue over time. So that's a really hard, first three things to think about, and there's plenty more as well.
Great, thank you. And I know we have a few other questions, but we'll have to respond to those a little bit later. And then finally, thanks so much to our presenters today, to the Wake Forest Institute for Regenerative Medicine and RegenMed Development Organization, to Alpha Med Press, and particularly to Bank of America for their support, but especially for the National Science Foundation Advanced Technological Education Program. Thank you for joining us today, and we look forward to seeing you in future webinars where we can bring to you the latest in regenerative medicine. Thank you very much.
Thank you.
Thanks, Gary.
Thanks, everyone.